Hello, I'm Les Bennett, and I have the pleasure of hosting a series of interviews with distinguished pharmaceutical scientists on behalf of the American Association of Pharmaceutical Scientists. The objective of the association is to put on tape, in a video format, interviews with the leaders in the pharmaceutical sciences who have made our field what it is today, and to get some insight into their background and their development, and certainly their thoughts on where the field is going. So today we are very pleased to have with us Professor Valentino Stella, who's the University Distinguished Professor of Pharmaceutical Chemistry at the University of Kansas. Val, it's great to have you here.
Pleasure.
Yeah. So Dr. Stella got his undergraduate degree in Australia. He is Australian, from the Victorian College of Pharmacy, and then came to the University of Kansas to receive his Ph.D. in 1971. Now, those of you that are really interested in a more in-depth view, the September 2012 issue of the Journal of Pharmaceutical Sciences is dedicated to Professor Stella, and there's a great deal of information probably beyond what we will say here today. But Val, give us a little history of, coming to the U.S., what was the story behind it, and what was your objectives, and what you were trying to do, and what you thought was going to happen.
Well, it's kind of an interesting story because I grew up in Australia of Italian parents, and I did very well at the undergraduate. My first year I got a lot of prizes. Second year I almost flunked out due to a number of reasons that we probably shouldn't go into. And then the third year I did well again. So I went to see the dean and I asked him whether I should consider graduate school. At that point, most people from Australia went to the UK, the Square in London or something like that. And he said, well, let me think about it. And so I came back a month later, and he said, you've been accepted at the University of Kansas. So I actually never applied. And so I ended up at Kansas because through Ian Pittman, who was an assistant professor with TAC when he moved down from Wisconsin to University of Kansas. And the year before, they had recruited Lang Fung from Buffalo, that's now at Buffalo, into the graduate. He did very well. So they decided to take a risk with a second Aussie, and as they say, the rest is history. But part of it was I just, I worked as a hospital pharmacist. I worked a little bit as a retail, and I wanted to be challenged by the sciences, and also always wanted to be a teacher. So that was a sort of a high. I wanted to be a high school teacher. My parents said you get a professional degree, and so I got the pharmacy degree, and as I like to tell the joke, is I end-run my parents because I got my Ph.D. and now I teach, so.
Yeah. Good. So you are a student of Professor Tatsuro Higuchi, and many view you as his, continuation of all the things that he did and the leadership role that he took, that you are someone who has taken on after him. So our audience is not going to be familiar with TAC. So I think you ought to give us a little history of that, and then let's talk about
Well, I don't think you should use my name at the same level as TAC. But TAC, as at least the older generation knows, he actually grew up in the Bay Area, ended up at University of Wisconsin, and got his Ph.D. at Wisconsin in physical organic chemistry with Willard. And during his time as a graduate student, he got to know
Joe
Swintoski. And Joe kind of got him interested in pharmaceutical sciences. And because he was Japanese-American, actually, when he graduated, he went to work for the Cleveland Rubber Company because of the war-related industry and being Japanese-American. And after the war, he actually was invited back and was on the faculty at Wisconsin. And effectively, as you know, Les, he's sort of considered the founder of the field of physical pharmacy. Many of us, if you look at the pedigree of many of the people, I think you studied with Sid Regal.
No, I didn't.
No, you were very close to Sid, and Sid was one of TAC's early graduates, and TAC was just so influential. I mean, we joke about it now, just because his memory is leaving us because Google doesn't search back far enough to some of that literature. So I've always sort of modeled myself after TAC in some way, not necessarily intentionally, but I think like TAC, I can do some things intensely for a period of time and I get bored, and so I go on to other things. And just like TAC, he sort of went in and out of different areas of pharmaceutical sciences. He had an amazing impact. And I hope that, you know, what I've done is at least, I don't, in no way am I anywhere near the influence that he's had, but I do feel I've carried on his legacy, and I think that's a fair assessment.
Right. You had a good year last year, 2011. You won the Higuchi Research Prize from the American Pharmacists Association and the Distinguished AAPS Distinguished Pharmaceutical Scientist Award. So I think you're in the same class.
Well, to put it this way, last year was a good year. Yeah. And actually, I've had an amazing personal life as well as professional life, and in fact, my wife and I were reflecting on it recently. There's very, very little that I would change, you know, what's happened to me over the years.
So you're probably, I think, best known for your impact in prodrugs. And, but you may disagree with me, but
I think there's three areas. It was prodrugs, the cyclodextrins, and drug stability. I mean, those are the three things that I've known for. Yeah.
So let's talk a little bit about the prodrugs, because I think you actually, that was part of your Ph.D. project a little bit, and tell us, you know, where it went and what's your vision and what you were trying to do. And
well, it really goes back to another one of the giants, and Sid Riegelman. Sid was a consultant at the time with Alza Corporation. And when I joined TAC in 1968, sometime after that, it was not my initial project, but sometime after that, Sid had suggested to TAC that there was a problem with injectable Dilantin, that if you injected too quickly, you would kill patients. Apparently there was a he was doing some, you know, that early 70s, he was doing some late 60s, early 70s, was sort of really the beginning of the heyday of some of the growth in the pharmacokinetic arena. And Les was trying to do some pharmacokinetics with Dilantin or phenytoin. And he killed a number of dogs, and the story goes. And a lot of it was when they did the autopsy on the dogs, they found this white powder in the lungs that is a result of too rapid injection of Dilantin injectable. And so TAC, about a year into my graduate work, said, you know, what do you think we can do for prodrugs of Dilantin? And my PhD dissertation sort of evolved into that. It was not what I started off with initially, but as a result of my PhD work, we did come up with a water-soluble, injectable form of Dilantin. It actually went into testing at Park Davis, but it turned out it had a cardiovascular effect. And so I learned a couple of things from that. One is you can make prodrugs. Two is the pharmacokinetics looks great, but you know the prodrug's also got to be non-toxic. And so that particular prodrug approach actually never made it past animal testing. And it was only a little bit later in the mid-70s that I had another idea, and that led to phosphenotoin, which is the first drug that I developed that went on the market.
And there's, since then,
Fospropofol and tenofovir, the Viriad.
Viriad, right, yeah. So I said best known for prodrugs. I think you are, but cyclodextrin, certainly Captisol is a big area there. And give us a little background.
Six products there.
Yeah, give us a little background.
That's another really interesting little story. I had, when Ani Repta left the University of Kansas to join TAC's company, I took over the NCI contract. We still have the contract. It's to do formulation work of experimental anti-cancer drugs. And you may remember this, but about 1985, 86, or thereabouts, a number of patients died that were giving paclitaxel, which was the cremophore ethanol mixture with the idiosyncratic histamine release. And with the contract, I was told at that point, don't develop any more formulations containing surfactants. Well, it turns out after the fact, as you know, we learned how to handle that side effect with the antihistamines and the steroids. But we were told not to do it. Well, it took out of our hands one of the major tools that we had to solubilize intractable compounds.
This is the NCI told you not to.
NCI told us not to do it, and in no uncertain terms. Basically, you know, we could do, you know, solubility through ionization. We could do co-solvents. at that point, we were not allowed to do prodrugs on that contract. And so I was looking for another way to solubilize drugs. And actually, the project kind of started with Arnie Repta. Arnie Repta actually had made some beta-cyclodextrin sulfate. And it was non-toxic, but it also didn't work very well. And so I had a graduate student from Victoria, Kansas, Roger Rajeski, and I gave Roger the task of saying, you know, I asked him, I said, why are cyclodextrins as a potential solubilizer toxic? And what can we do to change the structure so that we still retain the complexation but don't have the toxicity? And we had a very simple idea, and that was when Arnie had done the sulfate derivatives, we knew that putting charges on it at least got rid of the toxicity. We didn't quite know the nexus between solubilization and toxicity. And so the whole idea was that when the cyclodextrins are excreted in the kidney, there was a recording of an event in 1976 out of actually the Upjohn Company of beta-cyclodextrin crystals being seen in the kidney. And so we said, well, if we stop the uptake, if there was an uptake into the kidney tubule cells, by putting a charge on the molecule, you would get a renal excretion, but we shouldn't have the renal toxicity. And that was our simple premise. Roger made the, what we call the 6-sulfonate, absolutely non-toxic, but absolutely bound nothing. And so Roger was at a point in his dissertation where he said, you know, do we have a dissertation here? And the answer was not really. And so he came by my house, and I pulled out a 6-pack of Foster's beer. And by the end of that evening, Roger said, and to Roger's credit, I give him a lot of credit for this. It wasn't me, okay? Roger said, I think if we move the charge further away from the ring, we'll have the safety and still have the binding. That was Roger. That was not me. And I said, okay, you got 3 months. And 3 months later, we discovered Captisol.
That's fantastic.
Yeah.
So if you think it had been Budweiser instead of Foster's, it wouldn't have happened?
No, I don't know. I'm a bit of a beer connoisseur. I don't think I would have stoked that load for drinking Bud.
So let's get into the graduate students. You trained more than 40 Ph.D. students, and maybe had somewhere about another 90 visiting scientists or tutorial people in your lab.
95.
So let's get an idea of your philosophy of what's important. We've heard a little bit about interacting with Roger, but what's your objective? What do you want to do when you're training a graduate student, and what are you looking for when they come in?
Well, it's pretty interesting because I've only found out recently that a lot of my graduate students really feared me. I always thought I was a pretty nice guy, but it turns out I did not have a great reputation amongst the students, but I demanded good quality work, okay? And I also demanded, it was a philosophy that TAC actually had that I think I've captured a little bit, and that was that the student during their dissertation actually has to show at some point some truly problem solving. It may not be necessarily the solution to the problem that we set out to do, but along the way there had to be a personal growth in that they demonstrate that you're capable of of original thought. You know, it's not just a set of events that go along, and at the end you finish that work and you write it up and it's a dissertation. So I always had that philosophy with my students, but I also had it beyond their research. I also tried to work very hard with the students about being able to communicate, being able to write. And I had one student who was a domestic student who really could not get up in front of people and speak. And I said, you know, if you're going to be a scientist out there, whether it's in a company or academia, you know, you've got to develop those skills. And he had he ended up with a very thick dissertation because he was a very good researcher. But it was only at the point that I felt he was comfortable in front of a crowd, you know, that I felt he was ready to graduate. And yeah, he ended up taking quite a while to graduate. But I felt that that was an issue that he had to overcome before he reached the point of, I'd say, being a Ph.D., right? And so I do try to treat each student individually. And one of the things that was very difficult in my group is that I'd be doing stability work, I'd be doing prodrug work, I'd be doing cyclodextrin work, and I only have about maybe half a dozen dissertations that literally followed on from a previous dissertation. I get bored with projects easily, so I tend to just do things that I really like to do, and I've been fortunate enough over the years financially to be able to get the money to do things that are fun, you know, and as a result of that, I think the kids get a lot of energy from that, and at one point I had 25 students, 25 people working for me, and I knew I couldn't handle that, and I sort of brought it back and realized that I'm very good at working with, 6 to 7 students, and I had that most of my career. I had a great time. I still have a great relationship with those students, despite the fact that at some point in their life they probably feared me. But
So leading to the funding is also really something useful to talk about. We're actually having 3 drug delivery scientists being interviewed today. And all of you, and you especially, have founded companies. And I think, I think the dynamic, and I think it's worthwhile to tell our audience, your perspective, the importance of founding companies, how that has potential problems at the university, or how those problems are solved, and what is the issues that you have to pay attention to.
oh boy, I mean that's a 30-minute question right there. Well, maybe I should take just one of the companies and say why we did what we did, okay? And Cydex was the first one that we started, and it was the Captisol company that designed to market, sell, develop Captisol. We had developed Captisol, and there was a very interesting event that occurred. It was within the Center for Drug Delivery Research. We'd use Kansas Technology Enterprise Corporation finances to take the basic idea that Roger developed and then to bring it into what I would call commercial grade material. And that means scaling up, optimizing the system, really finding out what the edges were, what are the things we could do. And I was presenting it to my scientific advisory board, which was a bunch of VPs, directors of pharmaceutical that I knew from the pharmaceutical industry, and one of them was George Mooney, my second Ph.D. student. And I'm really literally presenting the story of Captisol for the first time. It wasn't named Captisol at the time, but the sulfobutyl ether cyclodextrins. And I'm about halfway through my presentation, and a yellow pad slides across the table, and the yellow pad had written on it, "Shut up, let's negotiate." And it's literally one of those moments we're in the right place at the right time. Pfizer was developing VFend, the antifungal drug, and Janssen had cut off their supply of hydroxypropyl beta cyclodextrin, and all of a sudden Pfizer's saying, what do we do now? And fortuitously, George is in this meeting and sees his new cyclodextrin. I mean, classic case of right place, right time. Well, then we started to negotiate with Pfizer for the right to license Captisol, and that negotiation took 18 months, and it was horrible. And I could tell you some stories about Pfizer lawyers, but I probably, for a mixed audience, I don't think it would be it'd be worthwhile. But anyhow, we decided that the university, we sat back and said, boy, if we're going to negotiate, if we're going to have to negotiate with every company, and it takes us 18 months, the university's not set up for that. And so it was on the basis of essentially taking it outside the university, not necessarily for financial, it was also for financial reasons and everything else, but it was really with the idea that universities were not set up to do this. And so we started Cydex Corporation. The initial, we actually hired Peter Higuchi, Tak's son, to write the initial business plan, and after he saw the technology, he said, I want to be involved with it, and so he was the first president and CEO of Cydex. And Peter did a really good job of getting the company launched. Other issues happened a bit later on, but that's another story.
But what I want to do is go back in the, so what's the university's reaction to this? See, what I'm trying to get at is you're a university professor, and
you know, we
are,
actually never had any problems with that, and have not had any problems with it. And the reason for that is pretty simple. TAC blazed the way with ELSA, and with Interex, and later on with Oriad Labs. And so we'd already, as a university, come to grips with that, okay? And I'd also come to grips with the idea that I'd really love to teach. I'd really love to work with students. And even though I think I'm a pretty good businessman, that's not where I wanted to spend my energy. And I also had, at that point, a fairly young family. This was 93, 94 in that time frame. So I had a 12-year-old, a 9-year-old, and a 6-year-old. And I was the track coach. I was their volleyball coach, and I was very heavily involved. So I did not want, so the reason we started, one of the reasons I didn't get intimately involved in the companies as much as other people have, I think, is because I really did want to keep my role as a professor intact. And that actually hurt us at times when trying to raise money, because very often when the professor doesn't want to be the innovator doesn't want to be involved with the company on a day-to-day basis, that's viewed as being not interested. In fact, it was just trying to maintain my personal life.
Have you encountered any potential conflicts in terms of what your students or postdocs are doing and the companies that are out there, and how have you approached that and solved it?
Well, that's a difficult one. In fact, I've written a little story on conflict of interest, and it really is a conflict of interest that you're talking about there, in that, for example, Roger's dissertation, he actually could not go out and interview. Because we were keeping it under our hat. We actually filed a patent on Captisol the week before his dissertation defense. So we never presented that work in papers or posters or anything, because that would be public disclosure. But Roger bought into it. I told him what the pros and cons were. We made a conscious decision to not publicize the work until after the dissertation was defended and the patent was filed. When we did a lot more of the development work, we tended to use postdocs. But once we had the patent filed, we could publish the work. I mean, we were a little bit careful on some issues, but basically it really wasn't a problem. And in all, really, the university was pretty good about allowing us to sort of delay publications, till after the work was filed and things like that. So I didn't. I think the biggest conflict I ran into, and it was the reason, I was the director of the Center for Drug Delivery Research for 11 years. And at the end of the day, I had to get out of that business. And the reason I had to get out of that is that I was using state money to fund research that was leading to products that my name was associated with, that was going to generate potential royalties. And therefore, basically, I was using state money to fund research. And the conflict was out the wazoo. The way we handled it internally was fairly straightforward. We actually had a board that was independent. And so I and others would propose projects. They would approve it. And so, therefore, technically, the board approved the projects. But after about 10 or 11 years, I just realized, the perception of conflict there was so bad that I ended up I got out of that particular position, handed over actually to Roger Rajewski, and he ran up until fairly recently when that organization was dissolved. Yeah.
So some outsiders may think of the University of Kansas and think of basketball, and Val's a big basketball fan. But I think pharmaceutical scientists think of the University of Kansas and think of it as probably the place where pharmaceutical sciences is probably the most prominent among the university. And at least I view that as that.
Well, I think actually, there's a handful of universities. UCSF is definitely one. We're second to you guys every year on NIH funding. So, we're like Avis. We're trying to be number one, but in a healthy way, very much healthy. I think that the reason that we are perceived, I think tax legacy was important there, and obviously the legacy that Ron has, and hopefully the legacy that I've contributed to has helped. And, you know, we've got a young man, Corey Birkeland and others that I think are going to be the next superstars at Kansas, okay? And one of the things I'm really excited about in particular is that I think through Tack's leadership and Ron's leadership and Ken Artis's leadership and Christian Schonheit's, we've done a really good job, I think, of keeping a breadth of faculty in ages. We're grooming people to take over leadership roles. And in doing that, we're able to sustain the department. And I don't know a lot of departments that, a lot of places, I think, you know, some of the other institutions that, you know, have run in where a lot of people retire at the one time. And as a result, it's then very hard to carry on the legacy, you know. So I think really the leadership that Ron had and Ken and Christian, and hopefully I've contributed in the background, not in the formal chairmanship or whatever. I feel that was very important. And we basically, one of the neat things, a lot of people ask me, you know, how come, you know, this is my 40th year at KU, by the way, right? And somebody said, why did you ask for last 40 years, or why did you stay at Kansas? Number one was KU basketball. Number two is I just love the people I go to work with every day, you know. And that's not an accident, right? It's how you hire. It's how you recruit people. It's how you retain people. It's how you mentor people. You know, so even people that came in and you say, yeah, I'm not sure how good a colleague he's going to be, through mentoring, that person has taken on the culture of cooperation that Tack set. Ed Smithman also said in medicinal chemistry. And by the way, medicinal chemistry is doing fantastic at KU as well. And so, you know, we have a nice cross-section of people, and being pretty high on the totem pole at KU doesn't hurt when it comes around to how funding is allocated. So we've been able to maintain our position.
Right, but see, that's actually the point I was getting to, that in terms of the reputation of the pharmaceutical sciences, I think KU is a wonderful example of how the pharmaceutical sciences really are viewed by everyone outside as really.
Well, I hope that's correct. I mean, we like to think that way, and I think that perception is correct. But if you really look at it, it came from Tack's leadership, came from Ron's leadership, including things like GPEN. You know, so, you know, you've had an amazing influence too, Les, and, you know, not only as a UCSF professor, but what you've done with AAPS. And so those are all legacies that, you know, we leave behind, right? And we want the legacy to be more than just a bunch of papers, you know. Just not to sidetrack you a little bit, but it's become very evident to me how important our field is to public health. You know, I look at the drugs that I've helped invent and develop, and I have a colleague who is a multiple myeloma survivor, and Chuck, a fantastic musician, jazz musician, and he's alive today because of Velcade, you know. And I was introduced to a woman, a secretary, admin person at KU, and I was introduced to her, and she's shaking my hand, and she wouldn't let go of my hand. And I thought, what's going on here? You know, I was having all sorts of various thoughts. And it turns out that her father was alive because of Velcade, right? Yeah. And so all of a sudden you realize that you know, the tenofovir and the anti-AIDS drugs and things that you've done and contributed to, just the huge medical impact you have. And it's not just the people like that. I've been fortunate enough to develop products that, you know, you can directly say are the result of your research. What's really kind of neat is every one of us contributes in our own way. You know, my work built upon Sid Riegelman saying to Tack, we need a Dilantin, safe injectable Dilantin. That wasn't triggered from Tack. That came from Sid. That got to me. You know, phosphenotoin ended up being triggered by a completely other end. Ian Pittman had made the hydroxymethyl beta cyclodextrin. He was looking at that from a solubility point of view for oral dosing, and I saw the hydroxyl group and say, oh, wow, there's a handle, I could put a phosphate group on it. You know, so I built, you know, my ideas built on the shoulders of everybody else. You know, Ian was not the inventor of phosphenotoin, but if it wasn't for Ron's work, I would never have gotten to phosphenotoin, So we all build on each other, and you know, I've just been fortunate enough to connect some of the dots towards the end, but all our work's really important, you know.
Good, thank you. So let's get to, you know, other things. You and Beth have been the chairs of United Way in Lawrence, and so let's talk a little bit of, you know, your responsibilities outside the university.
Well, my wife is a developmental and child psychologist, and she, many years ago, took on a task of, it's called the Citizens Review Board, which is an organization, a group of people that help judges work with children in crisis and how to handle, you know, separating a child from parents, you know, due to drug addiction or other things. And so my wife sort of drugged me into the whole area of the need for the people less fortunate than ourselves. And I was very active in our church, and we're very active in some other areas. I served monthly at the Link Kitchen, which is the nutrition kitchen, soup kitchen. But I never really got involved in the community in the sense of broader. And I looked at this and I thought, you know, I did a tour of all the agencies for United Way, and there's one seminal moment that just crystallized in my mind. I was in the basement of the homeless shelter, and the basement was where all the families were. And I walked in. Number one, it was horrible, the intrusion in their space. And then I saw a young man lying on a mat, which is where he slept, and next to him was an 18-month-old little baby. And they were sleeping there at 11 o'clock in the morning. And I said, hey, do something about this, right? And so I started getting, my wife's been on the board of United Way for a number of years. We were co-chairs of the United Way campaign last year. I also do fundraising for the university, but I also am on the local hospital endowment association board. So I don't like the word giving back. You know, that's a word that a lot of people use. Actually, that's not a word that I like to use. I don't think you should give back. I think you should give. It's not like giving back means you're paying someone for something else. No, there are people in need and we need to take care of them. And so I've actually come to the conclusion that, yeah, I can see a hungry child in some country, but, you know, Lawrence, Kansas, you know, in the breadbasket of the United States needs help. And United Way's doing a pretty interesting, I get pretty emotional about this. United Way's doing an interesting thing. You know, you think of a lot of the United Way agencies as places where if there's a rape victim, they can go get counseling and help, right? And I call that sort of the finger in the dike. You know, you have an emergency and you plug it up. And what we're doing in Lawrence and Douglas County is we're getting all the agencies to work together. So we have a strategic plan on education, self-sufficiency, and health. And we got those all those organizations that are part of that have to fit into those goals. So if we can keep kids in school, they're more likely to get a job. If they're more likely to get a job, they're more likely to eat properly and have good health and have health insurance. So I have a goal of making the dike stronger, not just plugging the dike.
That's great. So let's talk. Let's end up with the vision of the pharmaceutical sciences, of where it's going, and you know, your input and what's the next generation.
You know, I think actually that's I actually am troubled by that. I mean, it's troubled by how the pharmaceutical industry is handling itself right now, as you know. And I think a lot of the problems are self-created. Okay. But I tend to be a cup half full guy, and so I look at the pharmaceutical industry a little bit like what happened with the brewing industry, right? So, you know, at one point you had these breweries in every city, and then they all became parts of Schlitz and Budweiser and everything else. And so all of a sudden it's like the car industry. You didn't have the boutiques, right? And then we have the Sam Adams came along, and Sam Adams becomes a major brewery, you know, just as Genentech is becoming a major company, Gilead's becoming a major company. So we're in flux where we were condensing down, and then but that created a whole bunch of opportunities for the biotech and startup companies, right? And then they fit in. Some of those groups coalesced. Some died, you know, rightly so. So I think the pharmaceutical industry is just now sort of repositioning, getting rebalanced. I think that's one thing. I worry a little bit about the sciences. And I worry about it in the sciences for a very simple reason, is that, you know, right now, I think the biotech and the biologicals and stuff, I mean, they're in their heyday. That will continue to be a major player. And so, like UCSF, KU actually bought into that fairly early. So we began in 1983, 84 training people, not so for the traditional pharmaceutical small molecule, but training people through Ron, Russ Middow, David Vulcan, other people, Taruna Sihan, Jen Lawrence, developing training people for the biotech industry. But NIH funding started to really shift towards supporting biologicals, biology-related research, important for identifying different things. The problem we run into is there's the big void in how do you train people to handle small molecule, and who does that training? And where is that space going? You know, I like to. I tell a lot of companies, you know, we have a chemical space out here that's this size, and we know if a molecule fits within that space, we can formulate it and deliver it. Everybody's trying to stretch that space, right? And you stretch that space by coming up with being able to deliver more complicated molecules better. But, you know, it's like the quote, Star Trek, you know, to go where no man and woman has gone, right? So there's molecules out here that are just not formulatable, right? I mean, they're just wrong. The space is wrong. So how do we drag them back into here, right? And I think, I don't see a lot of creativity in that area. And one of the reasons is I think it's a hard funding area. So I've actually set up a whole bunch of fellowships at KU through royalty monies that we'll be able to fund, I'm hoping, somewhere between 10 and 15 4-year, full-ride, self-perpetuating, vested scholarships that I'm hoping will be able to maintain KU's strength in that field, because we won't have to depend on NIH or NSF for funding in that field. And so I decided that, hey, if we can't beat it, let's find another way to beat it. So that was one of my fundraising efforts. So
that's fantastic. So Val, it's been a great pleasure. Thank you for sharing the information with our audience. I know they will all enjoy it, and congratulations to you on an outstanding
Now we have
career.
to figure out how to do one for you.
They've done one for me a long time ago.
Okay. Thank you very much.
You.